So now we are going to learn about MHC class 1 epitopes prediction. Okay.
So for that we are using a tool called Net MHC
Net
Pan
MHC Pan
4.1.
4.1.
Okay.
Okay.
So Net
Net
MHC
MHC
Pan
Pan
4.1.
4.1.
Okay.
Okay.
So this is
So
the
this
one.
is the one.
So
So
whatever
whatever the
the
protein
protein
sequence
sequence
you
you have,
have,
you
you
just
just
scroll
scroll
down.
down.
You
You
can
can
see
see
the
the
submission
submission
page here.
page here.
Okay.
Okay.
So
So what are the
sequence
sequence
you
you
have,
have?
you
You
just
just
paste
paste
your
your
sequence,
sequence,
and we have to
and we
paste
have to
only.
paste only.
We can paste
We
all
can
the
paste all
sequences
the
also,
sequences also,
but it's
but
better
it's
to
better
paste
to paste
only one
only one
sequence,
sequence,
protein
one
sequence.
protein
Okay.
sequence. Okay.
And we have to
And we
paste
have
it
to
in the
paste it in the
FASTA format.
FASTA format.
Okay.
Okay.
So
So for
I'm
now,
just
I'm just
taking the
taking the
example
example
sequence.
sequence.
So whatever
So
the
whatever the
sequence you
sequence
have,
you have,
you just
you
paste
just
that
paste
sequence.
that sequence.
So whether you
So
have
whether you have
downloaded it from
downloaded it from
Uniprot, or whether
Uniprot,
you
or
have
whether you have
created your
created
own
your own
sequence,
sequence,
so that
so that
sequence you
sequence
paste
you
it
paste
here.
it here.
Okay.
Okay.
So now
So now scroll down.
So we'll
So we'll
leave all the
leave
things
all the
by
things by
default.
default.
And now the
And
main
now the
things
main
comes.
things comes.
So what
So
is the
what is
length
the
of
length
your
of your
peptide? So
peptide?
core
So core
peptide length.
peptide length.
Okay.
Okay.
So here we
So
are
here we
doing
are
this to
doing this to
identify the
identify the
epitopes.
epitopes.
That is
That is
class 1
class 1
epitopes. Okay.
epitopes.
These
Okay.
class
These
1
class 1
epitopes,
epitopes,
hopefully they
hopefully
bind to
they
the
bind
CD
to the
plus
CD
4
plus
cells.
4 cells.
Okay.
Okay.
CD plus
CD
4
plus
T
4
helper
T
cells.
helper
Okay.
cells. Okay.
So here
So
we
here
need to
we need to
select the
select
core
the
peptide
core peptide
sequence
sequence
of all
of all
sizes. Okay.
sizes. Okay.
So
So
basically we have
basically we have
size.
size.
The size of
The
this
size
core
of this
peptide
core
varies
peptide
from
varies from
8 to
8 to
14
14
amino acids
amino
long.
acids long.
Okay,
Okay.
select all
Select
here.
all here.
Okay,
Okay.
and here
And
select
here select
species. So
species. So
within
within
our
our
human
human
genome, we
genome,
have
we
different
have different
HLA,
HLA,
human
human
leukocyte
leukocyte
antigen
antigen
alleles. Okay,
alleles. Okay.
so within
So
this we
within
have
this we
many
have
alleles.
many alleles.
Simply we are
Simply we are
selecting
selecting
HLA
HLA
supertype which are
supertype which are
representative one.
representative one.
Okay.
Okay.
So when we
So when we
select this
select this
option, so
option,
we will be
so we will
having
be having
the
the
entire
entire
list of
list of
super type
super type
alleles.
alleles.
Okay.
Okay.
So just
So just
select all
select
these.
all these.
Okay,
Okay,
just use the
just
shift
use the
button and
shift
select
button
all
and
these.
select all these.
So once you
So once
select
you
here
select
all
here
these,
all
they will
these,
be
they will be
added over
added
here.
over here.
Okay, they will
Okay,
be
they
added
will
over
be added
here.
over here.
Okay,
Okay.
then
Then
scroll down.
scroll down.
So this
So this
option, this
option,
is the
this is
empty
the
one.
empty
This is
one.
the
This is the
optional one.
optional one.
So here
So here
threshold for
threshold for
strong
strong
binder. So
binder.
all the
So
values
all
we
the
are
values
leaving
we are
it
leaving it
default.
default.
Okay, it is
Okay, it is
0.5.
0.5.
Weak binder
Weak
is
binder
2.
is 2.
Filtering
Filtering
threshold
threshold
is
is
minus
minus
99.
99.
So we
So
can
we can
include
include
binding
binding
affinity.
affinity.
BA means
BA
binding
means binding
affinity.
affinity.
Include binding
Include
affinity
binding affinity
predictions.
predictions.
Then sort
Then
by
sort by
prediction score.
prediction score.
So
So
these two
these
options
two
we are going
options
to
we are going to
select.
select.
Then click on
Then
the
click on
submit
the
button.
submit button.
Okay.
Okay.
So once
So
you
once
click
you
on
click
the
on the
submit
submit
button,
button,
so we
so
will
we
get
will
a
get a
result
result
of this
of
type.
this type.
Okay.
Okay.
So this is
So
the
this is
result
the
you are
result
going
you
to
are
get.
going to get.
So here
So here
you can see
you can
this is the
see this is the
position
position from
this
where
peptide
this
is
peptide is
starting,
starting,
and this is the
and this is the
HLA
HLA
human leukocyte
human
antigen
leukocyte
allele.
antigen allele.
And
And
I think
I think
Geeta, you have
Geeta, you have
selected only
selected
one.
only one. Ikkada.
You have
You
selected
have
only
selected
this
only this
0 1.
0 1.
So that is the
So that
reason
is the
we have
reason we
all
have
0
all
1s
0
only.
ones only.
So
So
hope you
hope you
understood.
understood.
Here
Here
so we have to
so we have
select
to
the
select the
different
different
super type
super type
alleles,
alleles,
HLA
HLA
super type
super type
representative alleles.
representative
Okay.
alleles. Okay.
So you have
So you have
selected only
selected
one.
only
So that
one.
is the
So that
reason
is
we
the
are
reason
seeing
we are
only
seeing
one here.
only one here.
Okay.
Okay.
Okay,
Okay, now let us not deviate now as of now. So here
this
this
is
is
the thing we
the
have to
thing we have to
understand.
understand.
Okay,
Okay,
so all
so
this
all this
result, it
result
will be
it
very
will be very
clumsy here to
clumsy here to
understand. You can see
understand.
it
You
is
can see
not
it is not
organized.
organized.
Okay,
Okay,
so simply
so
what you
simply
do,
what you do,
so till
so
where we
till where
have
we
the
have
strong
the
binder
strong
till
binder
here,
till
so
here,
you can see
so you
here
can
it
see
is
here it is
WB, it is the
WB, it
weak
is the
binder.
weak binder.
So till
So
here
till
you
here
just
you
copy.
just copy.
Okay, copy
Okay, copy
everything.
everything.
So now
So now
open the
open the
Excel
Excel
workbook.
workbook.
Okay, open the
Okay, open
Excel.
the Excel.
Okay, you have
Okay,
either
you
you
have
have
either you
MS
have
Office
MS
or
Office or
WPS Office.
WPS Office.
Open the
Open
Excel,
the Excel.
paste the
Paste
entire
the
data
entire
here.
data here.
Okay,
Okay.
so once
So
you
once
paste the
you
entire
paste the
data,
entire
so
data,
this will
so
be like
this will
this.
be like
Now
this.
we need
Now
to
we
do
need to do
data
data
sorting.
sorting.
Okay,
Okay.
so
So
everything is in
everything
a
is in a
one
one
column only.
column
So we
only.
have
So
to
we have to
separate them
separate
into
them into
different
different
columns. Okay,
columns. Okay.
so for this
So
we
for
need to
this we
do
need to do
data
data
sorting.
sorting.
So click
So
on the
click on
data.
the
You
data.
can see on
You
the
can see
top
on
we
the
have
top we
option
have
called
option called
data.
data.
So
So
here
here
we have to
we
select
have
the
to
option
select the
called
option
text
called
to
text to
columns.
columns.
So
So
this entire
this
data is
entire
in
data
one
is in
column.
one column.
We have
We
to
have to
separate them into
separate them into
separate columns.
separate columns.
Each type
Each
should
type
be
should
separated
be
into
separated
separate
into
columns.
separate columns.
So text
So
to
text to
columns.
columns.
So just click
So just
on
click on
advanced
advanced
split.
split.
So the
So the
options will be
options will be
same
same
whether it is
whether
MS
it is
Office
MS
or
Office or
here
here
in
in
WPS Office.
WPS
Both of
Office.
them have
Both of
same
them have same
options.
options.
Okay.
Okay.
So just
So
click
just click
on.
on. Okay.
So
So
here
here
text to
text to
columns. Okay.
columns. Okay.
So just click
So
on
just click
text to
on text
columns.
to
Then
columns. Then
you have to
you
select
have
the
to select the
option called
option called
delimited.
delimited.
So here you
So
have to
here you
select
have
the
to
option
select the
called
option called
delimited.
delimited.
Then click
Then
on
click on
next.
next.
So here
So here
we can see
we can
between
see
each
between
and
each and
every thing
every
there is a
thing there is a
space.
space.
So here
So
you
here
select
you
the
select the
space.
space.
Okay, tap
Okay, tap
తీసేసి
తీసేసి
select the
select the
space.
space.
So ఇక్కడ
So
చూడొచ్చు.
ఇక్కడ
Now you
చూడొచ్చు.
can
Now
see
you can
here in
see
the
here in the
preview
preview
the entire
the
data
entire
is
data
how it
is
is
how
going
it
to
is
get
going to
sorted.
get
It is
sorted. It is
separated into
separated into
individual
individual
columns.
columns.
Okay.
Okay.
Then click on
Then
the
click on the
next button.
next button.
Okay. Then
Okay.
click on
Then click on
finish.
finish.
So now you
So
can
now
see
you
the
can see
entire
the
data
entire
has been
data has been
sorted.
sorted.
Okay.
Okay.
So
So
now
now
once the
once the
data
data
is
is
sorted,
sorted,
so the
so
lowest
the lowest
percentile
percentile
one
one
is the best
is the
one
best
actually.
one actually.
Okay.
Okay.
So for
So
better
for better
understanding, what
understanding
you
what
guys
you
do,
guys do,
so this
so
is the
this
entire
is the
data
entire
that we
data
have
that we have
sorted.
sorted.
Just paste
Just
it
paste
one
it
row below.
one row below.
Okay.
Okay.
So the
So
top
the
we need
top
the
we need the
headings, right?
headings, right? To
For better
for better
understanding.
understanding.
Okay.
Okay.
So take
So
this
take
top
this
column
top column
separately.
separately.
So if you
So if
sort
you
this,
sort
if you are
this, if
working
you are
on
working
multiple
on multiple
proteins,
proteins,
if you
if you
sort this
sort
first row
this
once,
first row
that will
once,
be
that
enough.
will be enough.
So this
So
now we
this
have
now we
pasted
have
this one.
pasted this one.
So simply
So
as we
simply
have
as we
done
have done
previously,
previously,
go to
go
data,
to data,
text to
text to
columns,
columns,
then
then
delimited,
delimited,
then click on
then click
next.
on next.
Here you
Here
select
you
the
select the
space,
space,
then click on
then click
next
on
and
next
finish.
and finish.
So
So
these are the
these are the
headings what we
headings
need.
what we need.
So you
So
just
you
paste the
just paste the
headings.
headings.
So now you
So
can
now
see
you can
here.
see here.
So this is the
So this
core
is the core
peptide and this
peptide
is the
and this is
complete
the
peptide.
complete peptide.
So
So
now
now
sort
sort
this
this
one
one
according to the
according to
core
the
peptide.
core peptide.
Okay.
Okay.
Once you have
Once you have pasted the data,
sort this one
sort this
according
one
to the
according
core
to the
peptide.
core peptide.
So here you
So here
select
you
this
select this.
if you
If you
put
put
the
the
cursor here
cursor
on the
here on
top,
the top,
okay,
okay,
you can see the
you can see
downward
the
arrow.
downward
So
arrow.
then
So then
this
this
entire
entire
D column
D
will be
column will be
selected.
selected.
Okay.
Okay.
So now we have
So
to
now
do
we
the
have to do
sorting.
the
For
sorting.
this,
For
go to
this, go to
data. You
data.
can do
You can
sorting
do
here
sorting
also,
here
even on
also,
the
even
home
on
page
the home
also.
page also.
Okay.
Okay.
So data,
So
here we
data,
have
here we
sort
have
option.
sort option.
So
So
custom sort.
custom sort.
Then click
Then
on,
click
here we
on,
have to
here we
choose
have to choose
expand the
expand the
selection.
selection.
So
So
sort.
sort.
So
So
sort by
sort by
values,
values,
core,
core,
values
are
like
like
just select
just select
values.
values.
Here
Here
click on the
click on the
custom list.
custom list.
and we have to
we
select
have
this
to
A
select
to
this
Z.
A to Z.
Okay.
Okay.
So if
So
this
if
eight
this eight
ABCs are not
ABCs are
available,
not available,
just you can
just
click,
you can click,
you can add
you
your
can add
own
your
thing.
own thing.
Okay.
Okay.
Then click on
Then click
OK.
on OK.
Add
Add
kotte.
kotte.
You can add
You
your
can add
own
your
data.
own data.
Okay.
Okay.
Then click on
Then click on
OK.
OK.
Then
Then
click on OK
click on
here
OK
again.
here again.
So now you
So
can
now
see
you can
the
see the
data is
data is
sorted.
sorted.
Okay.
Okay.
And you can see
this
peptide
peptide
is
is
repeated
repeated
13
13
times.
times.
So but we
So
are
but
going
we
to
are
take
going
it
to take it
only
only once
for our
for
work.
our work.
Okay.
Okay.
And
And
this second
this
one,
second one,
so this is
so this is
repeated
repeated
4 times.
4 times.
And we are going
And
to
we are
take
going
it
to take
only
it only
once.
once.
Okay.
Okay.
And next
And
this
next
is
this
not
is
repeated,
not
this one
repeated,
is
this
not
one is
repeated,
not
ఇది
repeated,
కూడా
ఇది
repeat
కూడా
కాలేదు.
repeat కాలేదు.
Okay. So
Okay.
this
So this is
not
not
repeated.
repeated.
Okay.
Okay.
And
And
sometimes
sometimes you can see there will be a
uh,
uh,
very
very
little
little
difference. Okay, ma'am.
difference. Okay, ma.
So both
So
of them
both
are
of them
same,
are same,
but here we
but
have
here we have
V and
V
another
and
place we
another
have
place we
W.
have W.
So
So
which is having
which is
major,
having which
majorly
is
repeated,
majorly
that one
repeated,
you can
that one you
consider.
can consider.
Okay.
Okay.
So just
So just
consider
consider
this
this
W1,
W1,
and you can see
and you
it
can
is
see
also
it is also
repeated 9
repeated
times.
9
It
times.
means,
It means,
but we are
but
going to
we are going
consider
to
it
consider it
only
only
once.
once.
Okay.
Okay.
So this is
So
only
this is
once.
only once.
So this is
So this
repeated
is
again.
repeated again.
Okay.
Okay.
సో ఇప్పుడు
సో
ఈ
ఇప్పుడు ఈ
రిపీట్ అయిన
రిపీట్ అయిన
వాటితోని ఏం
వాటితోని
చేయాలి,
ఏం
హౌ
చేయాలి,
వి ఆర్
హౌ
గోయింగ్ టు
వి ఆర్
టేక్
గోయింగ్
ఇట్
టు టేక్
ఫర్దర్
ఇట్ ఫర్దర్
విత్ దిస్
విత్
రిపీటెడ్
దిస్
వన్స్.
రిపీటెడ్ వన్స్.
ఓకే.
ఓకే.
సో
సో
ఫర్
ఫర్
బెటర్
బెటర్
అండర్స్టాండింగ్
అండర్స్టాండింగ్
విల్ డూ
విల్ డూ
జస్ట్ యాడ్
just
సమ్
add some
కలర్స్.
colors.
ఓకే.
Okay.
సో విల్
So
గివ్
we'll
సమ్
give
కలర్
some
టు దిస్.
color to this.
ఓకే.
Okay.
సో దిస్ ఈజ్
So
ఆల్
this
ద
is all
రిపీటెడ్
the
వన్.
repeated one.
సో దిస్ ఈజ్ ద
So this
కోర్
is the core
సీక్వెన్స్.
sequence.
సో యూ
So you
జస్ట్ పేస్ట్ ద
just
కోర్
paste the
సీక్వెన్స్
core
హియర్.
sequence here.
ఓకే. ఆన్ ద
Okay.
రైట్
On the
సైడ్.
right side.
And now
And now
you can see
you can
both
see
of them are
both of them
same.
are same.
And here we
And
have
here we
K.
have K.
So you
So
just
you
add
just
K
add
here.
K here.
Okay.
Okay.
And next
And
one you
next
can
one
see
you can see
this is
this is
starting from the
starting
Y
from the Y
and ending
and
with
ending with
D.
D.
Okay.
Okay.
So just
So
add
just add
D here
D
at the
here
end.
at the end.
Okay now.
Okay now.
And next you
And
can see
next you
this
can
one.
see this one.
You just see
You
where
just
is
see
our.
where is
Excuse
our.
me, sir.
Yeah, Divya,
Yeah,
can you
Divya, can you
understand or
understand
not?
or not?
No, sir. After adding color for the similar ones,
Okay.
Okay.
Okay, ma.
Okay.
can you repeat it once again?
Okay. Okay,
Okay.
Divya.
Okay, Divya.
So you can
So
see
you can
here,
see
this is
here,
the
this
core
is the core
peptide.
peptide.
So in
So
all
in
these
all
colored
these
ones,
colored
just
ones,
check
just check
where our core
where our
peptide
core
is
peptide
present.
is present.
And
And
some
some
additional
additional
residues will
residues
be
will
on
be
the
on the
left
left
side,
side,
some
some
additional
additional
residues will be on
residues
the
will be on the
right side.
right side.
So that
So that
residues we
residues
are
we
going to
are going
add
to add
to the
to the
sequence
sequence
which we have
which we
taken
have
here on the
taken
right
here on
hand
the
side.
right hand side.
Okay.
Okay.
So or else
So
we
or
can
else
see
we
here
can see
this
here
Y
this
and D,
Y and
AI,
D A
AL,
I
AY.
A L
So
A
this
Y.
is the
So
core
this is the
sequence.
core sequence.
So in
So
this
in this
below one,
below one
we have
we have
K
K
extra to
extra
the
to the
left side,
left side,
right?
right?
And
And
in this
in
one
this
we
one
have
we
D
have
extra
D extra
on the right
on
hand
the
side.
right hand side.
And now
And
in
now
this
in this
one,
one
37th row I am
37th row
seeing
I am
here.
seeing here.
So you can see
So you can
from
see
where the
from
core
where
is
the core
starting,
is starting
YN
YN
DAY.
DAY.
Okay.
Okay.
So the core
So
is
the core
starting
is
from
starting
here.
from here.
So this is
So
the
this
core.
is the core.
And the
And the
remaining
remaining
GNR
GNR
GK is the
GK
one
is the one
which is
which is
additional one.
additional one.
Okay.
Okay.
So it
So
means
it means
here
here
you have
you
to
have
add
to
this
add
all
this
these
all
things
these things
G N R
N R
G K.
G K.
Okay.
Okay.
And
And
this one
this
in
one
the
in the
38th row
38th row
you can
you
see
can
this
see
is our
this is
core
our core
sequence.
sequence.
And
And
the remaining
the
is
remaining is
NRGK. It is
NRGK. It
already
is
added,
already added,
so no need
so
to
no
worry
need to
about it.
worry about it.
Okay.
Okay.
And
And
this one, it is
it is
RGK. This is
RGK.
our
This is
core,
our core,
and
and
RGK is the
RGK is the
additional region that
additional
is
region that
already
is
added
already
here
added here
on this side,
on
so
this
no
side,
need to
so no
worry
need
about
to
it.
worry about it.
So this
So
one,
this
last
one,
only G
only
is the
G
extra
is the
one that
extra
is
one that
already
is
added.
already added.
So
So
by
by
combining
combining
all these
all these
ones,
ones,
we are going
we
to
are going
generate
to
only
generate
one
only one
sequence.
sequence.
So that is
So
the
that is
one
the
sequence
one
here
sequence
which is
here
on the
which is
right
on
hand
the
side,
right hand
which
side,
you have
which you have
like
like
written on the right
written
hand
on the
side
right
now.
hand side now.
Okay.
Okay.
So like this
So like
we are
this
going to
we are
do
going
for
to do
other
for other
also. So
also.
let us
So
do
let
for
us
one
do for
more.
one more.
So you can
So you
see
can see
this also will
this also
consider
will
into
consider
this
into
one.
this one.
So first
So
let's
first
give a
let's
color
give a
to it,
color to it,
and this is the
and this
core
is the core
peptide.
peptide.
Okay.
Okay.
So now
So
you can
now you
see
can
in
see
the
in the
25th row.
25th row.
So this is
So
our
this is
core
our core
peptide,
peptide,
and
and
CRRL is the
CRRL is the
residues which has
residues
to be
which
added.
has to be added.
So here
So
you
here
just
you
add
just add
CRRL.
CRRL.
Okay.
Okay.
And next
And
one you
next
can
one
see
you can see
this is the
this
core
is the core
peptide,
peptide,
and
and
RRL is
RRL
already
is
added.
already added.
And
And
27th row
27th row
RL is
RL
already
is
added,
already
so
added,
no need
so
to add
no need to
again.
add again.
So
So
28th one is
28th one is
also L is
also
already
L is
added.
already added.
But now
But
coming to
now
the
coming to the
29th one,
29th
here
one,
you see
here you see
this is
this
the
is the
core
core
peptide,
peptide,
and
and
RRL is on the
RRL
left
is on the
side,
left
which is
side, which
already
is
added,
already added,
but there is
but
one
there
more
is
R
one more
residue
R
on the
residue
right
on the
side.
right side.
So this
So
we have
this
to
we
add
have
now.
to add now.
Okay,
Okay,
capital R.
capital R.
And next
And next
31.
31.
So
So
31 is like
31 is like
this
this
is
is
our
our
core
core
sequence.
sequence.
RL is
RL
added
is
on the
added
left
on the
side.
left side.
R also we
R
have
also
added
we have
just now.
added just now.
So no need to
So no
add
need
again.
to add again.
So you can see
So you can see
similarly
similarly you
the
can watch
remaining
the remaining
31 and
31
32
and
also.
32 also.
So the
So the
residues, additional
additional
residues we have
residues we
already
have already
added,
added,
L and
L
R.
and R.
Okay.
Okay.
And
And this one
only R is
only
there,
R is there,
so that we have
so that we
already
have
added.
already added.
So like
So
this,
like this
we have to
we have
merge
to merge
the common
the common
sequences and
sequences
create
and
one
create one
simple
simple
sequence. Okay,
sequence. Okay,
single
single
sequence
sequence
for each
for
core
each core
peptide.
peptide.
Okay.
Okay.
So this is
So this
how
is
you
how
have to
you have
sort
to
the
sort the
data
data
and
and
merge the
merge the
core
core
sequences
sequences
based on the
based
core
on the
peptide.
core peptide.
Okay.
Okay.
So the same
So the
thing you
same
have to
thing you
do
have to
for
do for
this one.
this one.
So like
So like for
whatever
the
the example
example
we
we
have
have
taken
taken
here
here
for this.
for this.
Okay.
Okay.
So for
So
this
for
one also
this
we
one
are
also
going
we
to
are going
do
to
the
do
same.
the same.
And these
And
ones
these
which are
ones
only
which are
single
only
time,
single time,
we are going
we
to
are
take
going
them
to
as
take
it
them as
is.
it is.
Okay.
Okay.
So this we
So
are going
this
to
we are
take
going
them
to take
as
them
it
as
is.
it is.
Okay.
Okay.
So this is
So
how
this
you
is
have
how
to
you have
sort
to
your
sort your
data
data
for
for
your
your
protein
protein
sequences. If you have
sequences. If
one
you have
protein,
one protein,
so you are
so
going to
you
do
are going to
once.
do
If you
once.
have
If you have
four proteins,
four
you are going to
proteins, you are
do
going to do
four times.
four times.
So
So
additional thing,
additional thing,
what is
what is
missing in
missing
this
in this is
this
this
HLA type.
HLA type.
Okay.
Okay.
So this
So
HLA
this
type is
HLA type
missing.
is missing.
So
So
this work you are
this work
not
you are
going to
not going
publish
to
anyway.
publish
So
anyway.
don't
So
worry.
don't worry.
We can
We
ignore
can
this
ignore
HLA
this
column
HLA column
for your
for your
project.
project.
Okay.
Okay.
But if you are
But if
doing
you are
real
doing
research,
real research,
we have to
we have
consider
to
this
consider
HLA
this
column
HLA
also.
column also.
Okay.
Okay.
So
So there is
nothing special
nothing
about it.
special about it.
So suppose
So
you can
suppose
see
you can see
everything here
everything
HLA
here
is
HLA is all
zero
are
one
zero
zero.
one zero one.
Sometimes it will be
Sometimes it will be
0901.
0901.
So it means
So it
whenever you
means
are
whenever you
taking
are
this
taking this
sequence, this
sequence,
core
this
peptide
core peptide
sequence,
sequence,
it can be
it can be
identified by two
identified by
different
two
alleles.
different
That is
alleles. That is
HLA
HLA
A0101
A0101
and
and
HLA
HLA
A0901. Both
A0901.
the
Both the
alleles
alleles
can
can
identify this
identify
core
this core
peptide.
peptide.
Okay.
Okay.
So that is
So
what
that is what
is the
is the
conclusion part
conclusion part
here. What
here.
we are
What
actually
we are
missing.
actually missing.
Okay.
Okay.
So
So
if you have any
if you have
doubts
any
now, you
doubts
can
now, you
ask me.
can ask now.
Okay.
Okay.
So then
So then if you have
no doubts,
no
then
doubts,
you just
then you just
finish the
process
process
for this
identification of
identification of
MHC class
MHC
1
class 1
epitopes.
epitopes.
Okay.
Okay.
So thank
So
you
thank
both
you
of you.
both of you.
Hope you
Hope you
understood.
understood.